All right. Hi guys. Our presentation today is on human disturbances and bird diversity in the Athabasca oil sands region. Next, please. So for our project, we're using a regional scale data set from 2014 that's collected by Environment and Climate Change Canada. This data set we use for evaluating the disturbance from construction and oil sand activity in an Athabasca area. Data collectors recorded count data of songbirds sightings across multiple habitat types and human disturbance levels in northern Alberta. The dataset contains over 12,000 individual birds across 141 species. Sampling sites span gradient of disturbance intensities described by variables including human footprint level and noise level. Next, please. So we decided to do a quick literature review, and we found that overwhelming evidence that anthropogenic activity like land use change, land degradation, and human activity negatively affects biodiversity. In particular, the decrease in biodiversity results in a decrease in taxonomic and functional diversity, which refers to the type of species and their role in the ecosystem. There also is a homogenization of taxonomic and functional diversity, which means that ecosystems are becoming more similar to each other. This points towards a decrease in specialist species. While dominant generalist species are becoming dominant everywhere, while rare species are decreasing, because generalists are favored in areas with high disturbances. This also results in a decreased evenness where there is prominent disparity between dominant species and rare species. Next, please.
We decided to measure species richness and evenness to infer potential homogenization in the oil sands region. We hypothesized that increased human footprint level and noise pollution in the Athabasca oil sands region will decrease the taxonomic diversity of birds and species evenness within the regions. Higher levels of human footprint and noise should result in lower species richness and evenness. This would mean that there will be lower total taxonomic diversity and a decrease in rare species within the community.
So to start our analysis, we first modified the bird sighting data set to construct our response variables. Species richness, which is our representation of alpha diversity, was calculated by tallying the number of species per site. And then we calculated Simpson's diversity index for each site to be used in evenness calculations. And these evenness calculations were based on both the Simpson's diversity and the species richness, which we previously calculated. And we chose evenness measure because it is completely independent of species richness, unlike other diversity indices, so that we could isolate just the evenness. And it indicates the relative abundance of species in a community and how dominant the common species are.
For explanatory variables, we use human footprint levels and noise levels. They are both ordinal categorical variables, each with 5 levels, ranging from low disturbance to high disturbance. We converted both explanatory variables into numeric variables with levels 1, 2, 3, 4, and 5. These changes allow us to accommodate PCA analysis and GLS regression analysis, which is needed for spatial autocorrelation that will be discussed later. 4 sampling sites had to be removed because only 1 species was found within them, meaning that there was essentially no diversity and the evenness index could not be calculated. We also removed a data point that had NA for noise level. Furthermore, evenness values were log transformed to better fit a normal distribution. The sampling sites stretch over 4 degrees in longitude and 3 degrees in latitude. We calculated Moran's autocorrelation coefficient for both richness and evenness. And the results showed that there are moderate and statistically significant clustering among the sampling sites. Thus, a spatial structure was included in the statistical models.
For the model selection, we will ultimately select 4 GOS models to separately analyze the effect of human footprint levels and noise level on species richness and log Simpson's evenness. The selection is for accounting which of the 5 spatial structures is the most fitted. In total, we generate 20 models and look for the AICC values. The exponential spatial structures have the clear lowest AICC values across the 20 models, indicating that they are the best fit. Thus, we use these 4 models with exponential structures in the subsequent analysis.
We also did we also did a factor analysis and mixed data to determine the effects of each variable on the result. Next, please. So the factor analysis and mixed data showed that none of the human disturbance variables have significant influence on the variance in the data. We looked at 5 dimensions with the highest contribution, and dimension 1, or principal axis 1, explained 0.3% of the variation, and principal axis 2 also explained 0.3% of the variation. The 5 dimensions in total explained 1.4% of total variation. For dimension 1, human footprint level had the highest influence. Despite this, we determined that no factor significantly influenced variation. Next, please.
So the table shows the result of our analysis. As you can see in this table, all 4 models yield a p-value of more than 0.05, suggesting no relationship between human footprint and noise level, and species richness and species evenness. We also, next please. We also checked for the assumption of normality and homogeneity in variance. The model of noise affecting the species evenness seems to violate normality and homogeneity of variance assumption as shown by these 2 graphs. The QQ plot does not follow the line in both extreme ends, and the residual plot doesn't have a similar distribution. The other 3 models have a slight deviation from the assumption, but are still able to account for the analysis. Next, please. So ultimately, our results suggest human disturbance is not important in determining songbird species diversity. The models we use are not perfectly compatible with the data. Other models can still account for the different distribution other than the Gaussian distribution, which will better fit our response variable. For example, Poisson distribution would better fit the species richness as it is a type of count data. The assumption of GLS models can also restrict the analysis.
In addition, the noise level values may be confounded. Since most of the bird variations are based on bird calls or bird songs rather than visual identification, the intensity of noise will affect both the hypothetical response of birds and the ability of the observer to hear the birds at a distance. For instance, by looking at the detect type in the dataset, less than 3% of the birds were actually identified by visual confirmation, and that a noise level of 4, which is intense noise or over 60 decibels, the observers noted difficulty hearing birds more than 25 meters away and probably only detecting only the closest or loudest birds. Our analyses also don't take into account some variables in the data set, such as weather conditions or access point type, which describes, for instance, if the site was natural or if the site was on a pipeline or a seismic line, which could also account for some of the response in diversity. As seen in our box plots, there's a lack of visual difference between the human footprint level and noise level of different sites. And it's possible that songbirds are not as affected by human disturbance, either because they're highly mobile compared to other animals, or because they have a delayed response to disturbance and did not show changes in diversity at the time of observation. It's possible that human disturbance in the Athabasca region either lowers the diversity of other taxonomic groups or possibly affects the diversity of birds in later time points. But based on our analyses, songbird diversity is not affected by human disturbance.
Thanks for listening, everyone.
Thank you.